So still good morning to everyone. I'm Vladimir Meglič and I'm researcher at the Agricultural Institute of Slovenia, dealing with agrobiodiversity, and within that with let's say vast number of issues that agrobiodiversity is addressing, from evaluation of genetic resources to plant genetics and plant breeding. And okay. And today I will give you presentation on the pre-breeding, where we're gonna take a look at some basics, some examples, looking at the additional material. At the end, we'll have a small quiz and discussion, wrap up, and proposed homework. So it will be in the same manner as Adrian was presenting his information on genetic resources, and as well the information that was given at the beginning. So how to get the certificate, to fill in or to send the homework and so on. So that is pretty much the same as it was conveyed at the beginning. In any way, if there are newcomers, then at the end, maybe Petra will once again repeat what needs to be done and how it needs to be done, but you'll get all information as well through the slides. I believe that we're all aware of the climate change, and as Barack Obama has said, we're the first generation to feel the impact of climate change and the last generation that can do something about it. Yeah, that's definitely true. And as you know, that humankind is trying to alleviate the impacts of climate change on the in a global way, and definitely through the United Nations, and a lot of countries has signed that they will try to do everything to reduce or alleviate the global warming. Some countries might get out of that, but nevertheless, the majority of the humankind is trying and doing everything to alleviate that. Looking at the agriculture, there is a lot of global plans of action and different roadmaps how to deal with the climate resilient agriculture, or how to achieve a climate resilient agriculture. And FAO has a special plan, Green and Climate Resilient Agriculture, where they're addressing complex issues of the food security, reduced poverty, building resilience, greening agri-food systems, fighting the climate crisis, to reduce the greenhouse gases, and as well to reduce the impact of climate change. When we take a look at how the environmental outcomes of climate change has impacts on the agriculture. So as we know, and we are reading that every day and seeing the pictures global wide, what is happening from increased temperatures to increased flooding, to the fires, increased drought, the erratic rainfall that we experienced, increased storm intensities, and so on. All that has impacts on agriculture. And what we can see is the We are experiencing increased disease severity. We have, because of that, we have lower yields. We have lower nutritive quality of our food and so on. So these are only some of the examples what the impacts on the agriculture are. When we take a look at the, let's say, on our plants, we can see that plants are dealing with a lot of stressors. So we have natural stressors and the anthropogenic stressors. When we take a look at the natural stressors, we can divide them by abiotic and biotic. And from abiotic, drought right now is probably the most important stressor among the stressors that we need to and we are dealing with. Flooding, salinity, cold, heat, mineral deficiency, increased light, increased UV radiation, oxidative stress. When we take a look at the biotic stressors, we have from viruses, bacteria, fungi, weeds, herbivores, insects, competition, allelopathy, everything. From the anthropogenic stressors, we need to point out the herbicides or the pesticides. Air pollution is worse and worse, acid rain, fog, heavy metals, and so on. And on the right-hand side, you can see the impacts of all the impacts that plants have to deal with, as we had photosynthetic light, even gravity, photoperiod, humidity, wind, CO2, pathogens, and as well from the soil side. So it's soil quality, different parasites, water status, as we have said, mineral nutrients, toxic materials, and soil microorganisms are putting the pressure on a plant. And now, from our perspective, and the title of our presentation is the question that arises: how can the pre-breeding or breeding serve as a solution to the specific demand of organic agriculture in the area of climate change? So we will try to, or I will try to answer on that question. And we would start with the plant breeding and definition. And we would say that plant breeding is a human selection of the best plants from a diverse population. So we can say that the increasing number of people, climate change, new plant diseases and pests have always demanded the creation of new, more resistant and more productive varieties of existing plant species, or the introduction of completely new or wild species. So the main goal of plant breeding is to create new, more productive, more useful for humans and for the environment-friendly varieties. Well, we should not forget as well more useful for the animals, since a lot of plants are as well devoted for the animal feed. The process of breeding consists of the three basic parts, and we should say we are creating genetic diversity, selection of the best individuals from genetically diverse population, and realization of the variety. So that means testing and reproduction of the selected individuals, stabilization of genetic diversity, validation, maintenance, and distribution of the variety. So this is in only a couple of words what we do when we breed our plants or our crops. So We should say that since of the beginning of the 20th century, plant breeders have methodologically leveraged an ever-increasing set of knowledge, skills, and tools to add value to the plant genetic resources for food and agriculture, and they made crops better for the humankind. But some of the crop varieties might already be unsuited to the challenges and opportunities that our environment is, let's say, throwing at them. So what are the alternatives? How to achieve genetic gain in the absence of easily exploitable traits or genes? And how can genes be accessed to address this multiplicity of needs? So some of the questions you have got as well through the Adrian's presentation, and that was on the genetic resources. So significant efforts and investments have resulted in the collection, characterization, evaluation, documentation, and conservation and distribution of plant genetic resources. So we should say that most of the world's important crops are in gene banks, and these gene banks are reservoirs for different variations of the traits still used by farmers, traits that are not longer needed by farmers, traits that evolved but may never previously have been used by farmers, or traits that have not yet been discovered. So this is everything what we have in gene banks, and your last exercise looking at the CGN database is actually that. So they are the reservoir of the variability that you as a plant breeder would be able to use in your work. We have about more than 7 million accessions in over 1,300 gene banks all over the world. But unfortunately, these accessions are not used optimally in crop improvement. And this is because lack of documentation and adequate descriptions of collections, since we have a lot of accessions that has not been yet been evaluated. They are waiting for us to be discovered. So it's insufficient evaluation of the collections. There's limited input by breeders during documentation to understand what information would be most valuable. So this is as well one challenge that gene banks are facing, and they're trying really hard to understand what breeders are looking for and how the gene banks can help them to get that information to them. So there are accessions as well with limited environmental adaptability, accessible materials not always suited for the agronomic needs, and as well the adequate quantities of seed are not available in a timely manner. So that means that if there's only, let's say, some seeds, and even though that there's quality system in the let's say in a major world gene banks that are trying to get the adequate quantities of seed, so that if there is a demand, there is enough seeds for the exchange. Some of the gene banks might not have that yet in place, and for that some of the seed quantities are small, and if in case that you would need that accession, you might be waiting a year or two so that that accessions is the seed of that accession is increased. So the narrow genetic base of cultivars coupled with low utilization of genetic resources is the major factor limiting production and productivity globally. And to exploit this genetic diversity, pre-breeding offers a unique opportunity by introgression of desirable genes from wild germplasm into cultivated backgrounds readily used with minimum linkage drag. So this is something that every, let's say, pre-breeder or breeder would like to have. And this pre-breeding term was first coined by Charlie Rick in 1984, and Adrian has already told you a small story of this famous researcher and explorer. And at the end, I will touch to his work as well. And the pre-breeding is an alternative term that is used for genetic enhancement, and in recent times it has become an essential planned part of all breeding activities. It refers to all activities designed to identify materials that cannot be used directly in breeding programs and further to transfer these traits to an intermediate set of materials that breeders can use further in producing new varieties for farmers. So a little bit more of the definitions. So pre-breeding is defined by FAO as transferring of useful genes from exotics or wild unadapted sources into agronomically acceptable background or breeding material. And further, the Global Crop Diversity Trust defined pre-breeding as the art of the identifying desired traits and incorporation of these into modern breeding materials. So the pre-breeding is the transfer of or introgression of genes or gene combinations from unadapted sources into the breeding materials, including those that, although adapted, have been subjected to any kind of selection for improvement. So all goes from to get the set of genes or one gene or trait from wild or unadapted material into the adaptive one, so that could be readily used in your breeding programs. So pre-breeding is focused to enhance genetic variability in the germplasm, and improved germplasm can be readily used in regular breeding programs for cultivar development. And it aims to generate new base populations for breeding program through the use of a wider pool of genetic material. As well, it generates new base population for breeding program through the use of a wider pool of genetic material, and it has been successfully used in vast number of crops. So just to mention, rice, tomato, potato, soybean, cotton, maize, wheat, barley, groundnut, chickpea, and so on and so forth. And by transferring the genes from wild or exotic or unadapted species into adapted material, and with that, many cultivated varieties were improved for different qualitative or quantitative traits. Here's just let's say a small glimpse into the some of the crop species. Like beans, for beans, wild varieties are a potential source of novel alleles that can be exploited for the improvement of yield and other quantitative traits. In tomato, the introduction of the genes from Lycopersicum pinelii resulted into the raised level of provitamin A in the fruits by more than 15-fold. So these are things that a lot of researchers are looking for, and then as well plant breeders. In cotton, since 1970, there has been a lot of pre-breeding in different countries, And there was resistance to biotic stress, abiotic stress, and quality improvement as fiber quality was introduced into the cultivated background. There's are examples from groundnuts, pigeon peas, chickpeas, and so on, where they were improved by using the wild species for resistance to biotic stress and abiotic stress, and as well the quality improvement. At the end, there is example for the fruit species where the cultivated mulberry was crossed to and used different wild species for its improvement of the quality. So what would be the objectives of pre-breeding? Should say improve germplasm and associate genetic knowledge that enhance resistance expression and diversity, reduce genetic uniformity in crops through the use of a wider pool of genetic material to increase yield, resistance to pests and diseases, and other quality traits, identification of desirable traits or genes in their subsequent transfer into a suitable set of parents for further selection, improve parental stocks, which can be readily utilized within breeding programs, and improve selection methodologies, identify potentially useful genes in a well-organized and documented gene bank, designing strategies that lead to development of an improved germplasm that are ready to use in varietal development. So here we can take a look at the some of the difference between pre-breeding and traditional breeding. And on the left-hand side, we have what the pre-breeding does and what the traditional, on the right-hand side, what traditional breeding does. So pre-breeding is also known as a genetic enhancement, and traditional breeding is defined as sustainable plant breeding. So pre-breeding leads to genetic enhancement of germplasm, and with traditional breeding, we are trying to develop productive cultivars and hybrids. In pre-breeding, we usually leads to value addition. So we are trying to add some values for further breeding. It leads to broadening the genetic base of the population. And in traditional breeding, we're trying to develop improved cultivars with more narrow genetic base. The main breeding method is a backcross method in pre-breeding. In traditional breeding, all breeding methods such as introduction, selection, hybridization, or mutations are used. The end product of the pre-breeding is improved germplasm line, and in traditional breeding is a cultivar or hybrid that we put on the market. In pre-breeding, the end product is used as a parent for development of improved cultivars and hybrids, and in traditional breeding is a commercial variety, and it's used for commercial cultivation. In breeding, it involves adapted and non-adapted genotypes in the crossing schemes. And in traditional breeding, we try to use only adapted genotypes. Pre-breeding is really a long-term breeding program, or the pre-breeding program are long-term programs, and the breeding is a short or medium-term breeding program. Pre-breeding, it usually taken up by public sector or some of the public breeding organizations, and traditional breeding is both by public and private sector. In pre-breeding, we can as well look and we can maybe even try to use some kind of a decision tree when pre-breeding would be needed or useful, and when probably it would not be needed. So no pre-breeding needed if one of the following is available. So if we have commercially available, adapted, and acceptable varieties, advanced selections that are well adapted to the target environment, and gene bank accessions that are well adapted to the target environment. So all those are where there's not additional effort needed to include the genes from unadapted material. So therefore, the pre-breeding probably would be needed if gene bank accessions are not well adapted to the target environment, if closely related wild species are easily crossed with the crop species, or wild species that are less closely related and more difficult to cross. So these are, let's say, main examples where let's say, would be the pre-breeding would be needed. What are the classical approaches that are used in crop improvement with the help of plant genetic resources? First is introgression. And introgression is also known as introgressive hybridization. And it is the movement of gene or gene flow from one species into the gene pool of another by the repeated backcrossing of an interspecific hybrid with one of its parent species. And this introgression is a long-term process, and it may take many hybrid generations before the backcrossing occurs into the usable background. So it is transfer of one or more genes from exotic or unadapted or wild stock to the adapted breeding material. The other approach is incorporation, and this could be understood as a broadening of genetic base, and it refers to a large-scale program aiming to develop locally adapted population using exotic or unadapted germplasm. And the objective or incorporation is to produce new breeding populations that have very high proportions of unique exotic derived alleles in order to broaden substantially the crop's genetic base. Then we can use as well a white process. So when we have, let's say, two individuals belonging to a different species or different genera, and such a cross can be rarely realized in nature, and has to overcome a lot of barriers that are prohibiting the development of the fertile offspring. And we know that some white crossing has been used with some or with considerable success in some crops, and let's say as example, blight resistance in potato, rust resistance in wheat, insect resistance in rice. So there are a lot of there is a lot of work that has been done to use the white grasses to introduce the genes or traits of interest. As well, decentralized participatory plant breeding or participatory plant breeding is one of the means that plant breeding programs are using. And they're a little bit different than the let's say mainstream or programs that are established either at the private or even public plant breeding institutions, where we have help of farmers that are helping the plant breeders in different stages, creating variability, selection, and as well testing of the experimental cultivars. A decentralized participatory plant breeding program also functions with the same line, provided with some differences, like most of the process takes place in farmers' fields. The decisions are taken jointly by the farmers and the breeder, and the process can be implemented at a number of locations involving a large number of farmers evaluating different breeding materials. So this helps as well to look at the same or very similar germplasm at the different locations and looking at the adaptability of that kind of material along the different environmental conditions. Then we have as well market assistant breeding, that it's based on the molecular marker patterns instead or in addition to their trait values. And when we have those kind of markers available and they're conveniently co-segregating with candidate gene, the market-assisted selection or market-aided selection may improve the efficiency of selections of simple traits in conventional plant breeding programs. And that has been used more and more and more. And now with the advent of, let's say, better and more efficient molecular markers, this part of work is as well very important in the plant breeding or plant improvement or pre-breeding programs. We should say that pre-breeding acts as an interface of conservation of plant genetic resources and breeding. And this is a multidisciplinary approach and requires the following: collection of the underrepresented diversity informed by gap analysis based on taxonomic designations and ecogeographic information, along with a detailed passport data to both conserve the breadth of the diversity available in the wild and facilitate predictive trait mining based on the ecogeographic data. Coordinate evaluation and sharing of pre-breeding products across environmental conditions to better understand genotype-environment interactions. Improved information sharing of that information and feedback, especially with regards to genotypic and phenotypic data and the way they are linked between genetic resource conservationists, pre-breeders, breeders, and end users. And enhanced coordination between basic and applied science research communities. And we should say that there are several challenges that are associated with genetic enhancement programs, particularly with genes that are introgressed from wild species. And here we have listed some of the challenges that we could name. It's cross-incompatibility in interspecific crosses, stability barriers and chromosome pairing in hybrids that have restricted the access to genes from wild species into cultivated ones. The linkage drag, this is one thing that it's really important. Hybrid inviability and sterility, small sample size of interspecific hybrid population, restricted genetic recombination in the hybrid population, lack of availability of donors for specific traits like resistance to diseases, insects, and pests. Exchange and accessibility of cultivated species germplasm material has become difficult due to the as well legal restrictions like the IPR, so like intellectual property rights. And this has been in the last couple of years even let's say more let's say burning issue, as some of the genes are already patented and the material that is carrying those genes for that material. If you want to use it, you need to as well pay some of the fees. So this is as well one of the issues that is coming with the advent of new techniques and technologies into the plant breeding. So once again, pre-breeding is a highly multidisciplinary set of activities, and the core are the gene bank manager and the plant breeder, and they are supported by a large group of involving disciplines such as molecular biology, plant biology, entomology, physiology, soil science, and others. There is a greater likelihood of more complex issues for cross-pollination, such as timing of flowering, incompatibility, and irregular seed development, which need to be understood by the plant breeder. And pre-breeding is not a new science, but both the urgency for its use, as well as new opportunities for better identifying and using genes in exotic or wild materials, make it imperative to enforce our ability to do effective long-term pre-breeding and to establish and keep going the pre-breeding programs. We can further understand pre-breeding in terms of crop evolution and the opportunities of modern plant breeding. And if we take a look at the pre-breeding in the framework of the evolutionary and agricultural background, we should say that with few exceptions, humans selected our food and industrial crops from their wild progenitors in some cases as far back as 12,000 years. The combined forces of human and natural selection produce a wide range of genetic diversity. This heritable diversity gave crops the ability to adapt to different climates, soil types, photoperiod, and to meet the requirements for different management systems, market-determined quality traits, etc. Farmer and natural selection was responsible for nearly all the varieties of crop cultivated on the planet up to the early 20th century. When we take a look at the pre-breeding in a framework of modern plant breeding, we should say that the situation changed with the rediscovery of Mendel's laws, and from that, the birth of the science of genetics. Modern plant breeding was born when scientists began to understand how genetic information could be applied to improving crops. And scientifically based plant breeding came onto the scene and allowed dramatic changes in the ways the new varieties are produced. So if we take a look as well and then reflect on what Adrian was describing in the first unit in the morning, let's take a look what gene bank managers and what plant breeders do. And gene bank managers assure that the widest possible diversity is held safely in our ex situ collections under or even in situ under standardized protocols for the species in question. May also work to assure conservation in situ, but most don't have this responsibility. Gather and organize the passport data, including any available information about the traits and performance of materials were collected and the area itself through the GIS technologies. And this activity might be carried out also by the collectors who supply the gene banks with the collected material. Manage the information to minimize errors and to maximize the utility of accessions. Follow standards of regeneration and viability testing to minimize risk of loss. Characterize the germplasm for those traits that are environment stable and serve to reliably aid in identifying an accession. This can involve whole plant or molecular traits, which may or may not have any relationship to traits of economic importance. And this is as well the importance of the public institutions where the most of the gene banks are situated to preserve the genetic variability for the humankind. So what plant breeders do? Plant breeders evaluate a broad base of germplasm to identify sources of genes to combine into the varieties that will be superior to existing ones in some significant way. Their germplasm base of choice typically includes materials that are generally well adapted to the target region for the breeding program. The reason for this preference is that using a poorly adapted parent will bring a set of undesirable genes into the breeding population along with the gene or genes of interest. So we were saying that the linkage drag is, let's say, quite of a concern in plant breeding programs when using the unadapted material or material that resulted from the white process. Then the breeder will normally need to work for several additional generations to select out these undesirable genes while tracking and selecting for the desirable ones. And after extensive testing, including input from farmers at all levels, breeders work with seed multiplication, certification, and distribution systems to assure that farmers receive the intended benefits. So the essential role of pre-breeding is seen in cases when genes of interest cannot be found in adapted germplasm. And the joint roles for breeder and gene bank manager include evaluation for traits of importance, information management, study of the reproductive biology, additional multiplication to make materials available for crossing, and collaboration where crossing is difficult. So on this scheme, there is depicted the pipeline that goes from genetic resource to the farmer's seed. And we see the different stages of this pipeline where we have gene bank, we have pre-breeding programs and commercial programs or commercial or public breeding programs. And at the bottom, we see the work that needs to be done, that it's not completely self-sufficient, but has to have the overlap with some other work that needs to be done. So we see the collection, maintenance, characterization. So maintenance is overlapping collection characterization. So characterization and pre-breeding is overlapping with evaluation, and pre-breeding and hybrid production or the variety production is as well overlapping with the parent development. So at the beginning, genes are identified with unimproved genetic resources and their value is assessed. So with the evaluation, then these genes are introduced into the better agronomic backgrounds, tested, selected, and tested some more. So this is kind of like pre-breeding. And finally, these desirable genes are incorporated into materials having promise of commercial value. And this is the breeding. And the pre-breeding represents the middle segment of this pipeline. And as well, it depends on the traits of interest, how long this segment is going to be. Is going to be quickly solved, or the traits, genes, or let's say set of genes is more readily used, or is it hard to introduce into the usable background? Here's another similar scheme, seeing genetic resources, where we have cultivated type germplasm or wild varieties. We have the middle part of the pre-breeding evaluation to identify promising donors, hybridization to get to the trace into the well-adapted material, and development of breeding populations, and then it is handed on to the crop improvement, so either to the working collections or to the development of new cultivars or hybrids. So each of those has quite a time, and that is not set, let's say set in stone. How long will it take until we get to the end of these procedures? Here, once again, in a little bit different way, on the right-hand side, we have the simplified scheme of the pipeline that I have been already describing, where we have plant genetic resources and at the end of the released variety, and from the gene bank to pre-breeding and breeding, with characterization, maintenance, evaluation, parental development, and the varietal improvement resulting in a new variety. On the left-hand side, there's a more complex scheme of the genetic resources that we can as well divide into the different gene pools based on the ease of the crossing between them, where the primary gene pool is, let's say, the easiest, would be the genetic resources that are the easiest used in the pre-breeding and breeding programs, and further out we go, tougher it is to introduce the new traits or genes into the adapted germplasm. So the pre-breeding activities, once again, evaluation of wild relatives to identify promising accessions as donors, hybridization of the wild species to the well-adapted material, development of the pre-breeding populations or lines, evaluations of those lines across different locations and environments, and identification of the promising interrogations that have acceptable agronomic background. And as we have as well discussed, there's a lot of different science fields needs to be involved in all this work that we get the, let's say, result that we will be able to then push it over to the breeding programs, or to put together the pre-breeding populations that are enriched in variability, to put them into the working collection, and then to be handed over to the plant breeders developing new cultivars. Here we can take a look a path to the sustainable agriculture, integrating crop conservation and pre-breeding. So using the gene banks and pre-breeders or pre-breeding programs. And we have a first conservation of crops that preserve and safeguard diverse crop varieties, and they prevent the loss of critical genetic resources. We maintain or develop genetic diversity foundation. So this genetic diversity is the foundation of resilient crops, And these conserved crops maintain a diverse gene pool. We have rebreeding contribution, where we identify and utilize beneficial traits for crop improvement. And methods include genomic approaches and market-assisted selection, and enhancing the targeted traits, where we focus on the climate adaptations, disease resistance, stress tolerance, and transfer of specific traits from conserved crops to cultivated varieties. And we can say that conservation provides raw material, so gene banks provide raw material, and prebreeding is refining that material. Now, we will take a look at the couple of examples of the using the prebreeding prebreeding work in improving the varieties. And first one is the genome transfer of Solanum lycopersicoides into the Esculentum into the tomato using introgressions. And Adrian was talking about Charlie Rick, and this is actually his work, and I was fortunate and lucky enough to be part of this work. And I must say this was one of my favorite times of my professional life, that I could work together with Charlie, looking at the material that he has collected and developing that material further. So Solanum lycopersicoides is the perennial bush with really hard fruits. It's found in Peru and Chile, and usually even at the really high altitudes. It's self-incompatible and cross-pollinating species with 24 chromosomes. And it's possible to make F1 hybrids with tomato, but that is male sterile and incompatible. It carries resistance to abiotic stress, so low temperature, frost, drought, tolerance to mechanical damage, fruit firmness. So definitely for the canning industry, that was something that they were looking for for the combine harvesting and so on. It has as well resistance to biotic stresses, so to the viruses, bacteria, fungi, and insects. So this is definitely one wild species that was worth looking into it and to make the best out of it. And the hybridization scheme was developed to produce the inbreeding lines to be then further used in the breeding programs. And as we have said, that there was an issue with the male fertility of the F1 hybrids when crossing Esculentum, and they were using a cultivar WF36 with Solanum lycopersicoides. So after let's say thousands and thousands of crossings, there was one plant that was male fertile, and on the left-hand side, you can see that different accessions that were used that were promising, but only LA 2951 was promising enough with one plant that gave the male fertile progeny, and then that one was then back crossed to the VF36 to the tomato, and out of those from the backcross one, there was 281 total progeny. Out of those 200, like almost 300, only 1/10 was fertile. And again, it went to the further backcrossing, starting to with self-pollinations, and starting mapping of the Solanum lycopodium segments into the in a tomato background, and selecting homozygotes. Further work required as well some of the selfing and backcrossing to produce the introgression lines, and within that work there was a big selection against extraneous markers, and as well work to fix homozygous segments. And at that, let's say at the end of that, I came to the Charlie Lick's lab, and I was helping to build a series of the introgression lines that you have here for all the chromosomes, where with the where the dark spots represent the each chromosome, which each represent the Solanum lyco, so the wild part of the chromosome that is being fixed into the genetic background of the tomato. So with that, we have produced a series of introgression lines that, with covering the whole Solanum lycopersicoides genome, that was then given to the plant breeders, that they could use those in their further breeding programs. The next example is the pre-breeding of the Brassica oleracea and the responses to the organic agriculture needs. And that was done by our new colleague, Hajar Ben Amar. She got her PhD at the University of Catania, working on this, and as well she was part of the Horizon 2020 BRESO project dealing with plant genetic resources, breeding and seed production of the vegetable crops. And that was coordinated by Professor Ferdinando Branca. And she had 3 research lines. One was genetic diversity and germplasm characterization of the Brassica oleracea complex for collection. The second line was the effect of water stress on the variation of the biochemical profile of Brassica oleracea. And the third one was the evaluation of the Brassica based on agronomic trait, and she was looking at the glucosinolates in relation to water stress. And here's just briefly the results of each research line or part of her research. So looking at the biodiversity, she did the morphotyping and genotyping, looking at different traits and identifying and classifying the accession studies, and looking at the selection of the most polymorphic markers that could be further used in the breeding or evaluation programs. Looking at the water stress, the importance of selecting cultivars with efficient antioxidant system, as this trait is associated with higher chance of survival, improved performance under limited water conditions. So when we have a drought, there are cultivars that are better coping with that, and she was looking for those. And By considering morphological and biochemical traits as indicators of abiotic stress response, wild species and varieties, and Brassicola writes Gongilodes, Capitata, and Acephala, and Incrocio were identified as the most water stress tolerant among the studied cultivars. And here's the looking at the glucosinolates. She unveiled significant fluctuation in the glucosinolate content in roots and leaves in response to different irrigation regimes and genetic backgrounds. And the accumulation and profile of glucosinolates in plants are largely determined by genetics, but environmental and development variables also play an important role. So this is one thing what plant breeders need to be aware of, that not everything is geared by genes. So we have a lot of G by E interactions, and the promising candidates for breeding programs were identified in kales, broccoli, and cauliflower. Once again, the general conclusions. For the first part, genetic diversity exploration, a comprehensive examination of the Brassica gene pool, including wild varieties. She unraveled phenotypic diversity and genetic relationship using SSR markers that put the material evaluated in different groupings, and trying to help to understand the evolutionary history of those. She discovered specific biochemical markers that are linked to water stress tolerance and genotypes with exceptional drought tolerance. This is one thing that is then in a practical point, it's very useful in this kind of work. And as well, that was useful for getting information for domestication, plant breeding, and also for applications in food industries as potent natural antioxidant. So as well to use the brassicas for the not only for plant breeding, but as well to use them in the growing, in the organic growing of the of different crops, because of their exudates in the soil that help to alleviate different, let's say, issues with the soil pests and so on. And glucosinolates looks like that that insight into the how glucosinolate production and functions are influenced by drought, and that this is was the basis to explore the network of the glucosinolate biosynthetic genes and as well their regulatory mechanisms. So what we can say in conclusion, for crop improvement, sufficient genetic diversity exists in the form of land races and wild relatives, which carry several useful genes for cultivar improvement. However, utilizing of these resources in breeding programs is time-consuming and resource demanding. To overcome this, pre-breeding activities should be initiated to generate new genetic variability using promising land races and wild relatives for use by the breeders in crop improvement programs. Pre-breeding should focus on the continuous supply of useful variability into the breeding pipeline to develop new high-yielding cultivars with a broad genetic base. Pre-breeding should not focus only on increasing yield, for instance. Though pre-breeding is useful to enrich the primary gene pool for cultivar improvement, it is time-consuming and difficult as well. Further, linkage reg associated with utilizing wild relatives makes the pre-breeding activities much more cumbersome. And genomic-assisted pre-breeding will help to overcome the linkage reg and will facilitate focused transfer of useful genes from wild varieties for genetic enhancement. So this, I know it was a lot of definitions and so on, but this is something that I guess, and I hope made a little bit more clear what the pre-breeding is and what it demands and how it is linked to the genetic resources. So here we have some of the links and for the materials that as well I was using and that I would encourage you to use. So with the green star, I have marked the project on pre-breeding. So crop wild relatives diversity. So this is thing that you would, if you are interested in this topic, please take a look at that, as it gives a lot of useful information and interesting information, how to use crop wild relatives into the to enhance the genetic diversity, and how to use that as well for the in the pre-breeding and breeding programs. And definitely I would encourage you to, if you are interested in more detail on the pre-breeding or breeding or genetic resources, go to the FAO e-learning courses, where you can look and be part of, let's say, the global community that is learning from those materials. Even if you have, let's say, taken the courses, this might refresh your knowledge, and definitely will give you another, let's say, more detailed glimpse into the, let's say, problematics of the pre-breeding, genetic resources and breeding. And not only that, but as well other topics are covered with that e-learning programs. So I think we have now we have pretty much we're on time to finish when we should have been finishing. So this is the let's say one quiz with 7 questions. You don't need to send me the answers, as this is pretty much the simple answer, true or false. So please go through those and mark them for yourself, and then after 5 or 7 minutes I will come back, and so that means at 5 minutes to 1, so that's in 6 minutes, 5 minutes to 1, we will reconvene here. I will come back and I will show you the answers, or reveal the answers, and then we can go further on to the what we have learned today, maybe with a little bit of a discussion, and as well I will let you know about the homework. So here are 7 questions. Mark them for yourself, true or false, and then you will see how successful you were based when I show you the correct answers. So please, see you at 5 minutes to 1. These are the answers to the which is true or false. So except for 5, which is false, every other questions were true. So pre-breeding normally results in intermediate product that we have further crossing by the breeder. Yes, that's true, since definitely this is the part that you cannot release from the pre-breeding program. You cannot release, let's say, a variety, or very seldom. A pre-breeding project helps in building bridges between germplasm collection and plant breeders. That's true. Without close collaboration of the pre-breeding programs and gene banks, it would be really hard to deal with that, as the germplasm managers or the specialists for a certain crop, they have a vast knowledge on the behavior of different species of different crops, and they can give a lot of insight to the pre-breeder how to deal with the, let's say, obstacles when they arise, when they want to develop or incorporate the wild germplasm into the let's say, usable background. Question 3, the pipeline concept of plant breeding means that there is a constant flow, gradually improving germplasm for a series of stages of breeding. Yes, we have seen different schemes, how there is a flow of germplasm through the different stages, from the evaluation to the first crosses, back crosses, stabilizing those genes, and to handing them over to the plant breeders that they are further developing that germplasm. The risk factor for loss of genetic diversity is when breeders develop varieties that replace local land races. That's true. There is a lot of could be a lot of loss of genetic diversity when starting using the only let's say couple of varieties, and when they start dominating, then all the other local land races are being sometimes forgotten. But nevertheless, there are as well that's why as well we have germplasm collections or gene banks that are taking care of the local land races to put them into the gene banks, and as well the farmers that are aware of that, and they thrive and multiply and take care of those local land races so that they don't go into the vein. Question 5. One of the reasons for increasing needs to consider pre-breeding approaches is a need to shorten breeding cycle. That's false. Definitely pre-breeding cannot or is not intended to shorten the breeding cycle. There are different, let's say, tools and methods that do that, but pre-breeding is a long-term process that is actually usually not intended to shorten the breeding cycle. Pre-breeding is a way to reduce the vulnerability of crops when they have a narrow genetic base. Definitely, with pre-breeding, we try to widen the genetic base so that crops are more resilient and could cope with, let's say, changing environment that we have now, so that are more, let's say, in a way, to add the plasticity to the material that we are dealing with. And last question, pre-breeder needs to know whether a gene of interest is found in the crop itself or in the wild species. Definitely, because it depends on the biology of the crop, that so that the pre-breeder is aware of that, how to tackle, let's say, different obstacles when crossing the wild species into the background that is going to be then used. Just quickly on what we have learned today. Prebreeding is highly multidisciplinary set of activities. At the core are the gene bank manager and the plant breeder, and they are supported by a large group involving disciplines such as molecular biology, plant pathology, entomology, physiology, soil science, and others. There's a greater likelihood of more complex issues for cross-pollination, such as timing of flowering, incompatibility, and irregular set seed development, which need to be understood by the pre-breeder. Pre-breeder is not a new science, but both the urgency for its use as well as the opportunities for better identifying using genes in the exotic or wild materials make it imperative to reinforce our ability to do effective long-term pre-breeding. So the importance of germplasm resources for crop improvement is widely recognized, but their effective use in breeding can be complex, risky, and expensive. On the other hand, it can have very large rewards in terms of making valuable traits more accessible to plant breeders to address critical global issues such as food security and the face of climate change. Prebreeding is an opportunity to build a bridge that brings together the people who understand the scope and value of germplasm collections with those who need to introduce new traits into their cultivars. So with this, I would say that I hope I have, for those that were not that familiar with pre-breeding, that I opened the doors of this, let's say, beautiful part of the, let's say, plant genetics of the plant breeding that could be really tough, but at the end, really rewarding. And for those that you're already familiar with pre-breeding, that maybe I spiked some questions, maybe some thoughts, how to deal with the pre-breeding or how to use the material that is available on a global scale that you can use and in your plant breeding or plant enhancement programs. So at the end, I would propose a homework in the same manner as Adrian did. So if you could just send us a brief plan or pipeline on the pre-breeding activities on the species of your choice. Send that to my email and to Petra's, so vladimir.meglič@kas.si and petra.jelincic@ips-consulting.hr. For the questions, if you have any, please send us via or chat or email to Petra, and we will, as well Adrian has said, send them back to you in a nicely ordered, let's say way, so that everyone, even though that you have not asked any questions, you would have a, let's say, possibility to see the answers to the questions that have been posed. So with this, I would finish and I would say thank you. So we are a little bit over time. I don't want to go too much into your lunchtime, as you have as well another series of presentations or another series of very interesting practical presentations in this afternoon. So once again, thank you for your attendance. Thank you for being with us, and hopefully to see you all along until the first week of March with the last presentations. And of course, in case that you would go past the Agricultural Institute of Slovenia, don't hesitate to stop by.